Suppose in case of RBCs, no communication is required, they are individual cells and they are functions individually. But in many cases, there needs communication. As for example, in nervous system, the neurons connect with another neuron or muscle cell or gland cells, there needs communication. Similarly, in some cases, the cells come in close together and that form gap junctions. So, there are different types of communication. One is gap junction, where the cells connected to one another by means of these connections. You have read gap junctions. Substances can traverse from one cell to another without entering in the extracellular fluid, gap junctions. You are familiar with. It is important for the examination, gap junctions. Now, neural communication or synaptic connection, synaptic. It is a presynaptic neuron and it is the cleft and it is the postsynaptic neuron. Some chemical messengers are coming from this and it unites with the appropriate receptor, neural communication. Okay. Or it may be a muscle cell where there is liberation of neurotransmitter and there is appropriate receptor for this neural communication. And in some cases, the cells are situated close together and the chemicals secreted from a cell can diffuse to a short distance to affect neighboring cells. It is called paracrine communication. Paracrine communication. So, in the islets of Langerhans, there are different types of cells and hormones secreted from one cell do have effect on neighboring cells. Or in some cases, it is possible that it is affected by the cell, chemical said secreted by itself, autocrine communication, all the same thing. Suppose in paracrine, autocrine communication, autocrine. Communication. Suppose the cell is liberating some chemical and that is acted upon any cell in neighborhood. It has appropriate receptor or it may affect the cell that originally produced it. It is the autocrine and it is the paracrine effect. Yeah. And one important thing is the endocrine communication. Endocrine. Hormones secreted from cells of an organ coming into the circulation and carried by the blood to have effect either diffusely on selected target cells. Endocrine communication. But in this case it is locally diffuse. There is no need of the blood vessels. They may communicate. Paracrine or autocrine communication, locally diffuse. But in endocrine communication, there are the cells and they are coming, these secreting cells coming into the blood vessels and carried to have their effect in other cells. Endocrine communication. Thyroid hormone secreted from thyroid gland. Over this, there is thyroid cartilage placed and they are carried by blood to have effect almost all over the body. Insulin is secreted from the pancreas and it has extensive effect. Growth hormone is secreted from pituitary gland, anterior pituitary gland, and it has also extensive effect. In some cases, there are to some extent selected target organs where it may have its effects. And this is the endocrine communication. And there are other communications also, like juxtacrine communication. Juxtacrine. Some cells, they express transforming growth factor alpha. Suppose it is some cells are expressing that is the integral membrane protein. Suppose this is the transforming growth factor alpha, and another cell may have its receptor where it may have its effect. Submolecules. Suppose submolecules of this transforming growth factor alpha is situated over this cell, and another cell has appropriate receptor in this way, receptors, and they can come together and do have their effect, neighboring effect. Whenever there is localized growth, then this type of effect there. You are familiar with the integrins, cell adhesion molecules, and they do have their effect. They may communicate somehow from one cell to another. Cell adhesion molecules, integrins, they may have their effect. Communication. So in this way, there are communication between the cells. Gap junctions, it is present in the heart. In the heart, there are nexuses or gap junctions. Suppose in atrium and ventricle, they are separated by fibrous band. But if there is any ectopic focus, either in the atrium or in the ventricle, and it will propagate to everywhere, producing. শোনো, এখানে তো অনেক জায়গা রয়েছে, তোমরা কি খুঁজছো তুমি, ওখানে কোথায় যাবে তুমি? ওখানে স্মুথ মাসেল, suppose intestinal smooth muscle, this type of smooth muscle, they have gap junctions. So information reach from one cell to another by means of this. Similarly, synaptic junctions, it is a neuron, presynaptic neuron, postsynaptic neuron, or a neuron or muscle cells, and in this synaptic communication, neurotransmitters do have their effect. Paracrine autocrine communication, one cell may secrete some substance and it is locally diffuses to another to have their some effect. It is called paracrine, and even the chemicals, in some cases the chemical acts upon the cell that secreting that ligand, and it is the autocrine communication. Most important is the endocrine communication. A cell is secreting some substance that is coming into the blood and is carried to distant part, do have their effect. Juxtacrine communication, one cell expresses transforming growth factor alpha, and there are corresponding receptors of some cells that it can recognize, and they come together in this way. And it is this, there is this intercellular communication. Yeah. In this they communicate with each other. Now, how they affect them? Suppose the endocrine communication is important. How the ligand that is secreted an appropriate receptor, and it may have effect in different ways. There may be, in some cases, there is their effect on the opening of the ion channels. It may open or it may close. Suppose in case of skeletal muscle, the nerve is coming and it is liberating some chemical substance. In case of skeletal muscle, it is acetylcholine, and when acetylcholine binds with the appropriate receptor, muscle type nicotinic receptor, consists of five subunits, and there is opening of this channel, and there is entry of cations, mainly sodium ion, and there is a depolarization of the skeletal muscle cell, and ultimately the action potential is produced and it is spreading and do have its effect, muscle contraction. Or in some cases there may be closing of the channel, non-peripheral for the heart, and it may have its effect of closing the potassium channels. So, depolarization in this way is produced. Suppose in some cases, in this way ion channels, over the ion channels, and second is the action on the different types of kinases. Kinases. That is, they phosphorylate some proteins, kinases. Some may be threonine kinase, serine threonine kinase, or tyrosine kinase. Threonine kinase, or in some cases tyrosine kinase. And different types of kinases are there, and in that case there are and in this case there may be calcium dependent in some cases and cyclic nucleotide dependent. These kinases are cyclic nucleotide dependent kinases. Yeah. So, and of this it is important for the examination, it is the cyclic AMP dependent kinases, cyclic AMP dependent kinases, and also tyrosine kinase, cyclic GMP kinase dependent kinases also. Now we can discuss something about the cyclic, how it is the cyclic AMP is produced, and how it has its effect. And in this connection we have to deal with G proteins. And third important is this, that is the protein synthesis. Stimulation or inhibition, and in this case that is this thyroid hormone T3, T4, and glucocorticoids or steroids do have their effect in this way. Retinoic acid. My discussion will be to some extent incomplete because it is not possible to remember everything written in the book. But I will give some idea within this time, and you may go through books and have all the information, including those not covered by me. Now, cyclic AMP is important for the examination. It is produced from ATP, and in that case there are these term first messenger and second messenger, and it is a hormone action. It is a part of the hormone action. Yeah. First messenger, there is a hormone is first, and it has its appropriate receptor. Now you consider a cell. Hormones may be water soluble, and these water soluble messengers, that is the amino acids, amines, peptides, they are water soluble. They cannot easily enter into the cell. They do have their effect upon the respective target cells, and in that case there is these receptors, and there is specific receptors for them. And so in that case the hormone is the first receptor, and it is the and by means of which there is the ligand and it is the receptor. And by means of, and when it binds the receptor, then some messengers are produced. So hormone is the first messenger, or ligand is the first messenger, it is the second messenger. Second messengers important are these cyclic AMP, inositol triphosphate, diacylglycerol, calcium also can be included in this. There is these are these important second messengers. Suppose epinephrine acting upon the liver cells. Epinephrine liver cells, it has appropriate receptors over which it can act, and in response to this there is generation of cyclic AMP, and by the cyclic AMP there is increase in the activity or generation of the active phosphorylase, and phosphorylase for glycogenolysis and other effects, neoglucogenesis, or this. So that is the attempt to increase the blood glucose level. Now, how this ligand or the hormone is acting? We may take it as a hormone. It is the first messenger and this is the second messenger. When it binds, then there is generation of cyclic AMP from ATP, and in that connection, in most cases they are connected with the G proteins, GTP binding proteins or G protein, and they are coupled with, and this receptor, suppose you take the cell membrane, and when the appropriate receptor binds with it, and these receptors are peculiar, they have 7 transmembrane type of serpentine receptors. There are 7 transmembrane segments they have. They are coiled in this way, and there are, suppose in this way, N terminal outside, 1, 2, 3, 4, 5, 6 and 7. And when the ligand binds, suppose it is the ligand or the hormone, and it will bind with it, and in the third loop, and in which there are this connection with these G proteins. Beta, gamma and alpha. Heterotrimeric G protein with the third cytoplasmic loop, extracellular fluid, cell membrane, intercellular fluid, and this is the third loop. 7 transmembrane domain they have, and in the third loop there is the connection with this along with the plasma membrane. Then there is the alpha, beta, gamma, it is a heterotrimeric protein, and alpha is connected with GDP in the resting condition. In the resting condition, when there is no hormone, the third cytoplasmic loop contains this heterotrimeric G protein, that is the GTP binding protein, heterotrimeric 3 subunits, and when this ligand suppose binds with this in this position, when it is coming and it is uniting with this position, and then what occurs? Then this is converted into the alpha is united with the GTP, and beta gamma they remain combined. There is separation between them, and this has this activity. It is physiologically active, and it can open the ion channels in some cases, open the ion channels, and also this has also physiological activity upon ion channels also, and it is responsible for, it is a kinase activity, and it will in the membrane, it will cause, it will activate the adenylyl cyclase, and by means of which it causes the conversion of ATP to cyclic AMP, cyclic AMP. adenosine triphosphate, and 3' 5' subunit. Clear? 3' 5'. Shorthand, you can find out this type of structure. Adenosine, this type of structure, 3' 5'. In this case, they are in the prime sign. Prime sign, that is within in the ribose molecule. Clear? It is cyclic AMP, and it is converted from ATP. In case of ATP, at the 5' position, there is 1, 2, 3 phosphate moieties there, ATP, and it gives rise to this product, cyclic AMP, by adenylyl cyclase. And cyclic AMP comes within the cells and within the cytosol, and it has an effect. Adenylyl cyclase is bound to the plasma membrane. It is acted upon by the separated GTP alpha. Clear? And cyclic AMP is produced, and it will act upon the kinases, protein kinases. These protein kinases are present within the cytosol and inactive form. That is, they are R2C2 in this fashion. C2 means catalytic unit. R is regulatory unit. Suppose the cyclic AMP acts upon the kinase, that is the R2C2. It is the inactive product. R means regulatory, or C means catalytic. And when a cyclic AMP acted upon B, it will remove this regulatory group, remove them, and this C2 group will be free. And it is the protein kinase A. Or it may be several types. Protein kinase A. Clear? Protein kinase A, there is activation of some proteins by phosphorylating its serine threonine moieties. Typical example is phosphorylase. Glucagon, epinephrine act in this way. They have appropriate receptors, specific receptors, and they do have their effect in this way via these G proteins. Clear? And so these receptors, they are serpentine receptors, thousands of there. Similarly, there are thousands type of G proteins. In a good text, it is given, that is, it is 16, it is into 6 into 12. These are varieties. So you can multiply this to have this type of varieties. But everything is not present within the body, but it is possible. Similarly, there are thousands types of receptors. It is the N-terminal and the C-terminal. So you can think that this thing, the nature produces these appropriate receptors, and also this varying number of this type of G proteins. And they do have their effect via cyclic AMP. Cyclic AMP is a single compound, and protein kinase A is also, if not single, its number is low, and it has this effect. So specific effect can be produced by the nature. Cyclic AMP is mainly intracellular. Some amount may leak out of the cell. And when it is produced, now what occurs? Now after this alpha unit has intrinsic GTPase activity. GTPase activity. Then it will convert GTP again into, you consider it occurring in the cell membrane. Then alpha GTPase activity, again GDP. And this will come together, alpha, beta, gamma, they are reformed. Clear? বোঝা গেল? Heterotrimeric G proteins, and these proteins, they do have their effect. Initially they are in combination, and when the ligand binds with it, then there is GDP is replaced by GTP somehow, and then it is separation. It is separation of the alpha subunit with the combined beta gamma subunit. Beta gamma subunit it also have its some physiologic effects, but alpha subunit, it is primarily responsible for stimulation of the adrenal cyclase, that is at the present of the cell membrane, inner lamella, adrenal cyclase. 12 transmembrane domain structure cyclase, and it is cyclic AMP, then acts upon the protein kinase. It is the inactive protein kinase, and it takes away the regulatory unit, and then it will have catalytic unit, it will have its effect, and it phosphorylated different substances, enzymes like phosphorylase. What is the action of phosphorylase in the liver? তোমরা তো বায়োকেমিস্ট্রি অনেকটা দূরে এগিয়ে গেছে, হ্যাঁ? ফসফোরিলেসের কি কাজ বুঝতে পারছো? ফসফোরিলেস কি কাজ করে লিভারে? ফসফোরিলেস হচ্ছে গ্লাইকোজেনকে ব্রেকডাউন করতে পারে, বুঝতে পেরেছো? গ্লাইকোজেনকে ব্রেকডাউন করতে পারে। লিভারই একমাত্র জায়গা যেখানে ফসফোরিলেস, যেখান থেকে গ্লুকোজ সরাসরি বেরোতে পারে, অন্য জায়গা থেকে গ্লুকোজ বেরোতে পারে না, বুঝা গেল? লিভারই হচ্ছে একমাত্র অর্গান, অন্য জায়গা থেকে মাসেল থেকে বেরোতে পারে না। সেখানে এপিনেফ্রিনের সাহায্যে সেখানে ফসফোরিলেস অ্যাক্টিভেটেড হয়, এপিনেফ্রিনের সাহায্যে। এপিনেফ্রিন দু জায়গায় ফসফোরিলেসকে করতে পারে, অ্যাক্টিভেট করতে পারে। তার মানে inactive phosphorylase, when inactive it becomes phosphorylated, then it becomes active phosphorylase. And epinephrine has the capacity to do at the skeletal muscle and in the liver. Glucagon has its effect via the cyclic AMP in the liver, and when within the phosphorylase is activated, then from the glycogen glucose is produced. Glucose 6-phosphate is produced, and there is a phosphatase within the liver that is responsible for liberating glucose into the circulation. লিভারই একমাত্র স্ট্রাকচার যে গ্লুকোজকে সার্কুলেশনে আবার ফিরিয়ে দিতে পারে। In the skeletal muscle, this whatever is produced, it will not come into the circulation because it has no phosphatase. It is converted into, then by the Embden-Mayer pathway to ultimately pyruvate to be converted into lactate. Lactate can come into the circulation. Clear? And by neoglucogenesis, and that is activated also by epinephrine, can have their effect. Clear? In this way it is possible. And so this is this type, and whatever, and whenever is this formation, then how this reaction is terminated? Reaction terminated either by phosphatase, that is in this way the protein and protein phosphate is produced. Protein phosphate is produced by this catalytic unit. Either it can be removed by the phosphatase, or by phosphodiesterase it is converted into 5' AMP. Phosphodiesterase. 5' AMP. By phosphodiesterase that acts upon this phosphodiesterase. It is converted into inactive 5' AMP. So, this is the one mechanism, nucleotide. Now, calcium, how calcium ion do have that effect? Calcium ion do have that effect. Calcium has multiple functions: proliferation of cells, secretion, muscle contraction, various other type of activities everywhere. And calcium must be available within the cell in some cases. So, this contraction of the muscles can occur, secretion, proliferation, many other effects. And calcium concentration in the extracellular fluid is more and intercellular fluid is less. So, by means of some hormones or ligands, it is possible to increase the calcium ion concentration. And how it is possible? In case of calcium secreting, that is the hormone, suppose this type of G protein coupled receptors, G protein coupled GQ, it will bind, and when appropriate ligand binds with it, and then there is the activation of phospholipase C. Either beta 1 or beta 2 variety in this case, phospholipase C, and it will act upon PIP2, phosphatidyl inositol 4,5 biphosphate or diphosphate. It will, and it is in the inner lamella, it will act upon PIP2, phosphatidyl inositol 4,5 biphosphate. Phosphatidic acid, you have concept about phosphatidic acid. Phosphatidic acid kake bole? Phosphatidic acid, গ্লিসারলে থার্ড পজিশনে একটা ফসফেট যখন থাকে আর অন্য দুটোতে ফ্যাটি অ্যাসিড লেগে আছে, তাহলে সেটাকে বলে ফসফ্যাটিডিক অ্যাসিড। এবং তার সঙ্গে যখন ইনোসিটল লাগে তখন হচ্ছে ফসফ্যাটিডিল ইনোসিটল সেটা, এবং তার সঙ্গে যদি 4,5 পজিশনে যদি থাকে তাহলে পরে সেটা হচ্ছে ফসফ্যাটিডিল ইনোসিটল বাইফসফেট। এবং এগুলো খুব সহজ স্ট্রাকচার, তোমার বায়োকেমিস্ট্রি বইতে, ফিজিওলজি বইতে পাবে, বায়োকেমিস্ট্রি বইতে তোমার দেখে নেবে। এবং ইট ইজ কনভার্টেড ইন্টু PIP2 gives rise to IP3, inositol triphosphate, and diacyl glycerol, DAG, diacyl glycerol. Yeah. Inositol is hexahydroxy cyclohexane. Yeah. And it is then liberated. The structure is like this, you will find in your text. Inositol triphosphate, this is phosphate. This type of structure, this is 4,5 position phosphate, inositol triphosphate, 1,4,5 triphosphate, IP3, it is IP3, and it will be liberated within the cell, and it will act upon endoplasmic reticulum or other structures, and that is a liberation of calcium. Calcium, which stores calcium, and this calcium structure similar to that of troponin or so, calmodulin, and this IP3 liberation of the calcium was a liberation of the calcium, and calcium reacts with the unites with the calmodulin to have their effect. Its structure is similar to the ryanodine receptor. Ryanodine receptor, IP3 receptor is almost equivalent to ryanodine receptor in skeletal muscle. That we will discuss. I committed some mistake. Now this IP3 is the simple liberation of calcium, and this calcium concentration will rise, and it will in most cases unites with the calmodulin, calcium binding protein, calcium calmodulin complex. CAM, calcium calmodulin complex, and it have its kinase activity and also other activity. Clear? Calcium calmodulin complex. Calcium calmodulin is a structure, calcium binding protein. It has this type of structure, 4 calcium binding parts areas. Calcium, N-terminal and C-terminal, and 115, 115. It is trimethylated lysine and is highly conserved among species. K. Trimethylated, 115 position, which is conserved. And when calcium binds with it, it becomes an important kinase. And it will have its different effect, like suppose in smooth muscle, myosin light chain kinase, phosphorylase kinase, CA, calcium, there is calmodulin 1, 2, 3, these are the kinases produced. They have their synaptic function, protein synthesis also. Myosin light chain kinase is responsible for the contraction of the smooth muscle, that we may discuss if time is available. We will start with skeletal muscle. At least 2 classes are required to complete the muscle. Next week will be the muscle. If some part remains not covered, then you will learn yourself. Yeah. Now this IP3 is come into circulation, but DAG remains in the cell membrane, and it will activate the protein kinase C, different subspecies, 7 subspecies, protein kinase C, and it has the activity similar to that of the protein kinase A, that is produced with the cyclic AMP. Yeah, cyclic AMP. And inositol triphosphate responsible primarily for this, the liberation of calcium from endoplasmic reticulum, mitochondria, and in this way, liberate this. Now calcium ion concentration in the extracellular fluid is 100 nanomole per liter. 100 nanomole per liter. 100 nanomole per liter. That is equivalent to 10 to the power minus 7 mole per liter. It is the extracellular fluid, this concentration, calcium concentration, ionic concentration, this calcium. And it is the intercellular concentration, ICF, and ECF it is 12,000 more, ECF. It is it is multiplied by 12,000. So there is a tendency of calcium to enter from extracellular fluid into intercellular fluid, where its concentration is much lower, and there is also electrical activity, gradient is in its favor. So it now this IP3 that is produced, IP3 is terminated back into its original position. Suppose from PIP2, there is formation of IP3 and this diacylglycerol, and IP3 is then broken into IP2, inositol phosphate, in this way, and it will form the phosphatidic acid. And it will give rise to CDP, cytosine diphosphate, CDP, DAG, and they will ultimately form the phosphatidyl inositol. It is then IP2, IP phosphatidyl inositol phosphate, and then this PIP2. In this way, the cycle is completed. Yeah. Phospholipase C acts upon PIP2. Phospholipase C acting upon this, it is producing this IP3 and DAG, diacylglycerol. Inositol triphosphate converted into IP2 and inositol phosphate, and then it may convert into inositol, whatever it may be, and DAG is converted into CDP DAG, and with the inositol it forms the phosphatidyl inositol, and then PIP 4 phosphate, and then 5 phosphate. So phosphatidyl inositol P2 is produced. This is the cyclic way they are reformed. Yeah. In this way, the cycle is reformed. Calcium ion enters into the cell by different mechanisms. It may be ligand gated by hormone also, ligand gated. Some may be voltage gated, and some is store operated calcium channel, SOCC. Store operated. Suppose the calcium, if we consider calcium can entering into the cell by voltage gated calcium channel, ligand gated calcium channel, and also store operated SOCC, store operated calcium channels, and it is cell pushing it by means of this type of, it is actively transported calcium 2 plus and 2 hydrogen ion. In this way, it is actively transported, or there may be transported in this way, that is the sodium is coming in, sodium, and calcium is leaving. 3, and in this way they can come out. And there are also calcium ATPase is also actively transported in this way. And whenever, suppose the ligand gated channel, there is this, it will ligand gated hormone, then it is the IP3 that is responsible for the liberation. Suppose the IP3 is acting upon the endoplasmic reticulum and liberates calcium, and it is to be removed, and it is replaced by this same IP3 will have effect, so that the store operator channels, they become activated, and it will act upon this, so that it is replenish this calcium out, and this calcium is going out by these different types of mechanisms. My discussion is incomplete, but you will find in the textbook any inconsistencies and or incompleteness in my deliberation. Yeah. Now it is also another important thing. So it is something about this mechanism, but we cannot forget this type of activity. Protein synthesis, increase or decrease by means of this type of ligands. What are insoluble? Mainly that these corticosteroids or steroids in general, steroids do have their effect. Retinoic acid and thyroid hormones, they do have their effect, and they affect mainly the acting upon this nucleus and DNA. Genes, they have effect upon genetic effect. And how it is possible? Suppose they are this phospholipase C gamma is there also in calcium, liberation of calcium, and it is produced by tyrosine kinase receptors via the tyrosine kinase pathway. So there are crosstalk between this type of pathways. Clear? Now you come to some discussion about this corticosteroids, but steroids, and these are how they are effective. They are water insoluble. Steroids, suppose glucocorticoid, it is insoluble. It is entering a large number of cells, and this glucocorticoid, suppose cortisol, it will enter into the cell. It is a lipid structure, so it can easily enter into the cell, and within the cell it will unite with the appropriate cytosol. It is the nucleus, and it is a cytosol. It is a receptor. Normally the receptor is bound with some substances, suppose heat shock protein, or this substance is the cytosol. Heat shock protein causes it is inactive, and when cortisol enter into the cytosol and act upon this, then the heat shock protein will be separated. Then it will, this part, that is this receptor and cortisol combination, and heat shock protein will be separated, and it will enter into the nucleus. And when it will bind with this part of this DNA, hormone responsive element, and this type of receptors, whether it is nuclear or cytosol, it does not matter. Their behavior is the same, and these receptors have some, they are family of receptors, and the receptors has some basic structure. Suppose in case of cortisol receptor, at the C terminal, it is this cortisol binds with it, and this type was the DNA binding, cysteine rich receptors, DNA binding, and it is the ligand binding at the C part, and it is the amino-terminal part. Clear? And these are extremely, that is the specific part in which in some cases this cortisol can bind, and it is the DNA binding site, this receptor, and it is united with some protein, heat shock protein or so normally, so it becomes inactive. When cortisol binds with it, then there is change, there is this hormone responsive element. In case of T3, T4, this type of T3 receptors or estrogen receptor, their number do vary. their number do vary, but the basic thing is that. And this receptor then unite with the hormone responsive element, and in this case it is a CRE, cortisol responsive element of the gene. In the 5' flanks, it is there present, regulatory part. There, promoter region, basic, that is this, that part, that is gene, that part of the DNA. It contains at the 5' end, suppose the 5' end, that is the regulatory part, and then there is this the regulatory part, and it is the promoter region, and then there is the and this exon, intron, exon in this way, and this is the terminal part, poly A part in this way, C prime. This regulatory part, in this case, it is called hormone responsive element, and in which this binds. There is this DNA binding site, and it is the DNA binding site that unites with this responsive element at the regulatory region. And where most cases there is the activity of this genes, increase the activity. Yeah. Zinc fingers in this case. There is this, that by means of zinc fingers. This part contains 16 rich zinc fingers. In this way, this part, suppose it is the zinc, 16, this type of structure. This zinc fingers, by means of which it will bind with the cortisol responsive element, and that is increase in the transcription, or in some cases there may be suppression of transcription. In most cases there is increase in the transcription. So what is formed initially produced is the pre-mRNA, and pre-mRNA is converted into mRNA, and mRNA will come into the cytosol, and it then binds with the ribosomes. Ribosomes may be within the cytosol as free ribosomes, or it may be attached with the cytosolic surface of the endoplasmic reticulum, rough surface endoplasmic reticulum. And when they are produced, and they may come into appropriate, it may be an active enzyme, they can have their effect. They can have their effect. In this way, their number or their amount activity will increase. That is the enzyme responsible for neoglucogenesis, they can increase and do have their effect. And it is the mechanisms. Similarly, other substances, vitamin D metabolite, that is the calcitriol, it also has. Estrogen has its effect. It will directly receptor within the nucleus. By the same mechanism, it will have their effect. They do have their effect in appropriate target cells. Yeah. And it is the basic structure of these receptors. Whether it is nuclear or cytosol. At the C-terminal end is the ligand binding site, and in this case it is the DNA, more proximal to the N-terminal is the DNA binding site, 16 reach. And this part is relatively, that is the less specific also. These are basic structures. Their amino acid numbers are different. T3 and other receptors, and they have their same basic structure is the same. And they will appropriately bind with, there is this responsive element. Cyclic AMP case, then cyclic AMP, in that case pKa can enter into the nucleus, and it can phosphorylate some proteins called CREB. CREB. Cyclic AMP responsive element binding protein, and it can also increase the transcription. And there are in nature there are many types of receptors, and in some cases the ligands are not known. They are called orphan receptors. So our knowledge is incomplete. In the textbook, they declare that it is complex and everything is not known. And my knowledge is also much poor than those textbooks. There are 5 minutes remaining, and another important is cyclic GMP mechanism. From GTP cyclic GMP is produced. Cyclic GMP by guanylate cyclase. GTP cyclic GMP is produced. Guanylate cyclase. And this guanylate cyclase, and in that case it may be membrane bound, where there are these receptors and ligands. In this case it is the ANP, atrial natriuretic peptide. And for it there are A, B type of receptors, A and B type of receptors. And also gastrointestinal peptide, guanylin and E. coli toxin receptor, they behave in this. It is the membrane bound guanylate cyclase. But within the cytosol, there are guanylate cyclase. They are soluble. These are membrane bound, and it contains the structure is like the N-terminal end that bears with this. That is the ANP, and this type of guanylate cyclase activity in this tyrosine kinase and guanylate cyclase activity. But in some cases, like NO, it is entered within the cells. NO, nitric oxide, or endothelium derived relaxing factor. It enters into the cell, and in this case guanylate cyclase, then it is soluble, and it contains the heme, and it causes the activation or generation of cyclic GMP, and the cyclic GMP has also activated numerous kinases, GMP kinases, and they do have their effect, either in relaxation of the smooth muscle or other type of mechanism. Sodium reabsorption also in case of kidney tubule. My discussion is grossly incomplete, but I think that you have to go through books to have complete knowledge.